Hello friends, Ace Botane, next episode. In this episode, what we'll be discussing is molecular basis of genetics, part 3. And what all topics we'll be discussing? Almost 6 topics: search for genetic material, transforming principle, biochemical characterization of transforming principle, genetic material is DNA, properties of genetic material, DNA versus RNA. Then sixth one is RNA world. Seventh part, as usual, 5 theory question, 5 NEET model question with the key. So I will share this screen for convenience. I think screen A. So we can proceed. So first item is under 5.2, search for genetic material. So in connection with search for genetic material, some historical events is better to know. So Charles Darwin, here we can find photograph for scientists, and here the time in which they live. Charles Darwin, 1809 to 1882. G.J. Mendel, 1822 to 1884. Frederick Meister, 1844 to 1895. Hugo de Vries, 1848 to 1935. So when we analyze the years in which they live, it's very interesting to identify all our contemporaries. So all these great scientists, they have important contributions. But still, it took a long time to establish DNA was the genetic material. And some important experiments that led to prove DNA was genetic material is our next discussion according to NCERT. So we can proceed to experimental side. So many experiments will be there in NCERT. For theory point of view, NEET point of view, what we have to remember is scientist involved in it, then experimental organism involved in it, experimental chemical involved in it, what is the purpose, what is the aim, and what was the result. This way you can make a table for different experiments. It will be easy for you when you discuss or when you prepare for the exams. So we can start with experiments. In experiment side also, some important scientists and their years has been given. Conjugation, Totem and Lederberg, 1946. Transduction, Cinder and Lederberg, 1952. Replica plate experiment, you may study in evolution. Esther Lederberg and Joshua Lederberg, 1952. Transformation, Frederick Griffith, 1928. Then transformation, biochemical aspects, Avery, McLeod, and McCarthy, 1944. One gene, one enzyme, that is presently not in syllabus still. Beadle and Totem, 1941, in a fungus called Neurospora crassa, very famous fungus. Bioclassification will be knowing it, Neurospora crassa. And there are two types of organism, prototroph and auxotroph, original type and variant type or mutant type or associative type, etc. Just introduce certain scientist name and last one, organism as well as two terms. Now we can proceed to, I think first page is clear. 4 scientists we can find here, respectively, Darwin, Meister, that is Mendel, Darwin, Mendel, Meister, and de Vries. Now we can proceed to second page, transforming principle. It is connected with the famous scientist, Frederick Griffith. Photograph you can find, all details you can find here, 1928, British bacteriologist. And finally, giving a guess that DNA may be the genetic material. But the nature of genetic material not defined from these experiments done by Frederick Griffith. Now, experimental part, it is Streptococcus pneumoniae, bacteria that causes pneumonia or pneumococcus. 2 types, S3 and R2. S3 means smooth, capsulated. Covering will be there, that is shining also. Chemically, that cover is made up of polysaccharide or mucus. S3, that can cause pneumonia. They are virulent forms. Now, R2. R2 means it is rough, non-capsulated. They lack mucus covering. They are non-pathogenic or non-virulent type. They will not cause pneumonia. So the 2 bacteria, familiar, these are our usual questions. Then larger organism was a rat or mice. Now different combinations of S3 and R2 were injected into mice and checked what happened to mice. And this is how we can summarize it. S strain injected into mice, mice will die. R strain injected into mice, mice will live, survive. S strain heat kill injected into mice, mice will live. Fourth one is most important one. S strain heat kill plus live R strain. This mixture injected into mice where mice dies. So what the conclusion is, the heat kill S strain bacteria converts live R strain bacteria into live S strain or virulent form, by which rat will die. This conversion may be due to transfer genetic material from heat-killed S strain to live R strain. But still, even though this experiment is done, biochemical nature of genetic material was not able to define by this experiment, but giving a clue, that's all. That is regarding second page. Now we can proceed to third. I think idea is clear. Now, biochemical characterization of transforming principle. That is our next topic. It is done by three famous scientists. Name and photograph you can find here. Avery, MacLeod, McCarthy. 1933 onwards, they started the experiment, but reached a result in 1944. See, among scientists, before this experiment, different views are available regarding the genetic material. One prevalent view at that time was it is protein. But later, the scientists like Avery, MacLeod, and McCarthy in 1944, they repeatedly done experiments in vitro and finally proved genetic material is DNA. So a confirmation, not fully, still we got an idea DNA may be the genetic material. What the experiment part they done? See, they purified chemicals like proteins, DNA, RNAs, etc. From where? From heat-killed S cells. And observe, that is, which one could transform live R cell to S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. That is the term transformation, converting. And this part is given in NCERT. Page number 85, reprint edition, March 20. That is the essence of the experiment. And the diagram you can find, a simple diagram. So experiment to prove that DNA is the transforming material, Avery, MacLeod, and McCarthy. 4 combination you can find. These were injected. So culture medium plus RNA of S type, no change is caused. Culture medium plus proteins of S type, no change is caused. Then culture medium plus DNA of S type. And there is an enzyme called DNase. So that can digest DNA, no change. Nothing happened to it. Now culture medium plus DNA of S type. There we can find no enzyme. So we can find here some bacteria may be transformed into S type because enzyme absent. So DNA will work and converting our bacteria into S. Simple experiment, but it is a clue that gives the DNA will be the genetic material. And after that, we can find a line in NCERT, DNase and DNase. What is the difference? That is I wrote here. Theory question and that's question. DNase means enzyme that can digest DNA. That is what happened here. So DNA will be digested. So it will not be transferred. Here no DNase. So it can cause disease or conversion. Plural of DNA is called DNase. Plural of DNA is called DNase. Plural with S. Other one is DNase. I think this line clear. It is important for exam. Now we are further proceeding to, I think that page is clear. Evidence from experiments with the bacteriophage. More confirmation that DNA is the genetic material. But in NCERT, we can find a line, unequivocal proof. That is almost like an ultimate proof. For what? To show that DNA will be the genetic material. This experiment is otherwise called blender experiment. That's an important term. Question will be there for theory and NEET exam. Blender experiment done by. Unequivocal proof for DNA's genetic material is done by. And 2 famous scientists, Hershey and Chase. Alfred Hershey and Martha Chase, 1952, photograph given here. Purpose is to show that DNA is the genetic material without any doubt. That is otherwise called unequivocal proof. Here also organisms identify, we should know, and what all chemicals they use. So all items are given here. So virus, T2 bacteriophage, that will infect E. coli. Viruses, when we study bioclassification, you will be remembering viruses usually have 2 parts. One is DNA. Other genetic material, that is, other one is protein. So 2 chemicals. So scientists can easily check whether DNA or protein is the genetic material. Now, phage DNA labeled with the chemical is important. Phosphorus 32, DNA with the phosphorus 32, we know DNA contains phosphate. Protein code with S35, protein sulfur. So P32, S35, important question. Radioactive phosphorus and radioactive sulfur. That are the materials needed. Now, these are the two scientists. Now, regarding this area, sensory diagram, again important. So originally from today's episode, we can anticipate 2-3 questions, NEET model as well as theory part, important area. So in this experiment, blender experiment, 3 steps are basically involved. Purpose is finally unequivocal proof that is DNA is the genetic material. And what are the 3 steps? One is infection, other is blending, and third is centrifugation. And infection, what happens? So here both types of label, these are the 2 types of labeled phages, NCERT diagram, were allowed to infect bacteria. In one case, it will be sulfur, other case, it will be phosphor, that way. So that is infection part. Both types of labeled phages were allowed to infect bacteria. Now, blending. So bacterial cells were agitated in a blender. Purpose is to break the contact between virus and bacteria. Organism we know, bacteria and virus. So attachment, that should be broken. And that is done by an instrument or blending procedure. That is second step. And third step is centrifugation. So after blending, virus particles were separated from the bacteria by spinning them in a centrifuge. So 3 steps, infection, then attachment broken, and separation. And here we can find the experimental part. So very interesting, very simple when we refer the diagram. So we can proceed. So these are the bacteriophages. There we can find first one, radioactive S35 is the label. Protein, there we can find. Here in DNA, radioactive P32, labeled DNA. Because DNA contains phosphorus, then protein code contains sulfur. That is why S35 to this one, P32 to this one. I think idea clear. After that, infection, blending, everything takes place. And in the last, you can find here, no radioactivity detected in cell because it is connected with the code. While here you can find radioactive P32 detected in the cell. So first one here, no radioactive S35 detected in cell. Cell we cannot find it. S35. While here, radioactive P32 detected in the cell because it is connected with the DNA. Now what happened to this sulfur? After infection, blending, centrifugation, radioactive S35 detected in the supernatant area because it came out separated. While here we can find no radioactivity detected in supernatant because it is connected within cell. So I think idea clear. 3 steps, infection, blending, and centrifugation. This is the famous NCERT diagram and experiment, Hershey and Chase. Aim is unequivocal proof to show that DNA is a genetic material, otherwise called blender experiment. Now, properties of genetic material, I think it is clear. Properties of genetic material. So after a series of experiments, it is established that DNA is a genetic material. But after some time, scientists identified in certain viruses RNA is a genetic material like TMV, QB, bacteriophage, etc. Naturally, in majority of the organisms, most of the organisms, we can find DNA as a genetic material. While RNA usually perform the functions like messenger, adapter, then catalytic functions. So automatically, what are the properties needed for a chemical to consider it as a genetic material? NCERT clearly says it, theory question, NEET model question like odd one, etc. 1, it should be stable, no doubt. Chemical and structural should be stable. It should be able to produce its own replica, otherwise called replication. It should have the ability or to mutate, slow changes when needed and leads to evolution. It is able to express itself in the form of Mendelian characters. So for above the criteria, when we consider it, DNA will be the most suitable. While in RNA, we know second carbon atom there will be OH, makes it more unstable in comparison with the DNA. Second carbon atom OH is there. DNA, that O is absent. Presence of 5-methyl uracil, that is otherwise called thymine, in DNA gives more stability. So RNA become more unstable, faster rate of mutation will be there. Those viruses with RNA genome can directly code for protein synthesis. As a result, it can easily express the character. So evolution or change in mutation will be more in RNA virus. We know corona. Now comes the last area, that is RNA world. See, idea RNA world first proposed by Alexander Rich in 1962. Term RNA world coined by Walter Gilbert in 1986. So what is that idea RNA world? RNA world reflects the importance of RNA, clearly giving some clues for it. Why RNA is important? Some suggestions are, it is the first genetic material, essential life process like metabolism, translation, splicing, etc. linked with or evolved around RNA. Transcription, synthesis of mRNA from DNA. Then it can act as a catalyst. Ribozyme is an RNA-derived enzyme discovered by Tomsek and Sidney Altman 1980. RNA can be genetic material in some viruses. RNA is unstable. DNA came from RNA with the modification. So there are some important aspects why the term RNA world. I think page is clear. Now, when we enter into next area, there is a line or a usual question. DNA is more preferred for storage of genetic material since it is more stable. So storage of genetic material, better one is DNA, more stable. While RNA is more preferred for the transmission of genetic information like mRNA, etc. So these two lines important for exam point of view. Then as usual, 5 theory question, 5 NEET model question with a key. I think idea clear to all of you. So what all things we learned in this episode? We can just rewind it, starting from molecular basis of genetics inheritance, third one, 6 topics plus 7th one question for important scientists, search for genetic material and what is the contribution. And starting with the experimental part, again, some common experiments. Then transforming principle by Frederick Griffith, how the transformation takes place. Then after that, conclusion. In this experiment, still they are not able to define which will be the genetic material. Then biochemical characterization of transforming principle, that is by 3 scientists, Avery, McLeod, and McCarthy. Again, that experiment, that is enzyme DNA is with it and without it, how transformation takes place. And finally giving a clue, DNA will be a genetic material. Then again, unequivocal proof, that is given by blender experiment, Hershey Chase. DNA is a genetic material, virus, E. coli, then P32, S35 chemicals. And finally, 3 steps, infection, blending, centrifugation. And what happened? NCERT diagram, important. We can find S35 detected in supernatant, while P32 detected in cell. So radioactivity detected in supernatant S35. Here, no radioactivity detected in supernatant. P32 will be in the cell. That is how it was established. Finally, DNA will be the genetic material. What are the properties of genetic material? What are the conditions, criterions? What is the role of RNA? Then came RNA world, Alexander Rich and Walter Gilbert, important functions of RNA. And DNA is more preferred for storage of genetic materials. It is more stable. RNA is more preferred for the transmission of genetic information. After that, 5 theory question, 5 NEET model question with the key. That is regarding 3rd episode of molecular basis of inheritance. If you like this episode, subscribe all, like it, share it, comment on it. We can meet later with another episode. Thank you. Take care. Bye.